Hello friends. So in today's lecture, in the video series, we are starting the experiments which proves DNA as a genetic material. So we all know that three experiments were carried out to prove that DNA is an original genetic material which conserve and which transfer one strain to another, one mother to their offspring. Now the three experiments first was in 1928. Was given by Griffith. The second was in 1944. It is carried out by three scientists, Emery, McCarthy, and MacLeod. The third and last experiment was carried out in 1952. Just 8 years after this, was given by Hershey and Chase, which used the radioactive compounds. Now the three experiment was carried out to prove that DNA is a genetic material. So first we start with the Griffith experiment. It is also known as transformation experiment. Now we experiment and take a strain of Streptococcus pneumonie, which cause pneumonia. At that time, pneumonia is a very hard and death-causing disease. So he took two types of strain, the strain S and strain R. The S is named for its cultural characteristic because when it grows into nutrient medium, it gives a smooth appearance, and while R gives rough appearance. In S strain, it is a virulent type of strain, while it is a avirulent. Now it contains capsule, a polysaccharide capsule, while R strain do not contain any capsule, so no capsule. Now why R strain is avirulent and why S strain is virulent? So as we know, the S strain contains a polysaccharide capsule. So when it enters into any host cell, the macrophages or any APC or any phagocytic cell, when ingest this, because of this capsule, he can accept the phagosome or phagolysosome. While the R strain do not contain any capsule, that's why it do not escape from the phagosome or phagolysosome. That's why it is easily killed. That's why it is avirulent. Now the experiment was carried out into the mouse. Now how was the experiment? Let's see. It's a very easy pioneering experiment for proving the DNA as a genetic material and also the transformation of genetic material via transformation. Now, first he took a S strain and injected into the mouse, a healthy mouse. Now, after some days of incubation, the mouse was killed due to the effect of S strain and creating of pneumonia. Now, after this, we inject R strain into the mouse. After some incubation time, the mouse is still live and no infection was found about any pneumonia. Now then what he do? He give heat to the S strains and killed. So heat killed S strain and injected into the mouse. And after incubation time, the thing was changed. The mouse was still live. Due to it is a killed, so it do not produce any capsule, and that's why it is killed. The cell membrane of this cell is killed and disrupted. Now what the trick is do is take a heat killed S strain and mix it with a R strain, a live R strain. Now this mixture it injected into the mouse, and after the incubation time, the thing was changed and the mouse were killed. Now the question is why it is killed. As we know, the heat kill strain cannot kill him, and R strain is also avirulent. So what was happened between this? So mouse is killed. Now this is the real principle of transformation. That when these cells are mixed up, this is the S strain. Now S strain is a disruptive. That's why all the genetic material and all the cell constituent are coming outside the cell, and that's why by the transformation the DNA, exogenous DNA, is taken up by this R strain, and the Vir genes, which is virulent gene or capsule producing gene, is inserted via homologous recombination in R strain, and R strain become virulent. So this was the reason that why mouse is killed. So this was the first experiment carried out by Griffith to prove the transformation principle, and proving that there is some genetic material which present which cause any infection, which is responsible for any phenotypic and genotypic manifestation. So I hope you understand. Thank you.